Hi everyone, welcome back to the next video of the course. We will continue our discussion of fitness landscapes and how experimentally people try and work out how is evolution working in the context of these adaptive walks being studied on the analogy of fitness landscape. And just as a recap of what we did in the previous 1 or 2 videos, we discussed this paper by Daniel Hartl and co-workers where we compared 5 mutations in a single gene, and the effect of these 5 mutations was that it increased the MIC by 1000 fold. The increase of 1000 fold in MIC was accomplished via these 5 mutations, and we saw that transitioning from the strain which had a lower MIC to the one which had this 1000 fold MIC, there were 120 different paths or the order in which these mutations were acquired, which we could take to go from one strain to the other. And out of these 120 paths, if we used MIC as a proxy for fitness, only 18 were adaptive or permissible in a Darwinian sense, where natural selection will keep on increasing the MIC or fitness after acquisition of every subsequent mutation. But more important than that, and perhaps more surprisingly than that, 102 of these 120 paths were not accessible in a Darwinian sense, which meant that in between in these 102 paths there was a fitness peak that one encountered, which meant that you were trapped in a local maxima and you wouldn't be able to reach the 5 mutations. So while this makes protein evolution unpredictable because we wouldn't know that when we would get trapped in a local maxima, and this local maxima is accomplished because of something called sign epistasis, which we discussed 2 videos ago. So epistasis has this characteristic of making paths inaccessible. Sign epistasis does that. But it also means that instead of worrying about any of the 120 paths that this mutational pathway could have taken, I only have to worry about the 18 which were permissible in a Darwinian sense. So that was the first paper that we looked at. We will continue our discussion of the second paper, which was published in 2011 in Science, and the senior author in this is Tim Cooper. And just as a small recap of the experiment was as follows: that we have E. coli which is being evolved in an environment which has low amounts of glucose as the only carbon source that is available for the cells, and E. coli is being evolved in this environment for a long time. Obviously, mutations are going to get accumulated in the bacteria that are in this flask, and these mutations are going to be the ones which make E. coli fitter in the environment that they are dealing with. So after acquisition of 5 mutations, the fitness, if this was the ancestor E. coli, and let us say this was the genome of this ancestor E. coli, then after acquisition of 5 mutations, let us say this is evolved E. coli and this is its genome. This evolved genome has 5 mutations which are spread in 5 different parts of the DNA and is associated with 5 different genes which are concerned with 5 different functionalities inside the cell. If the fitness of the starting genotype, let us say fitness of starting genotype let us call this 1, then after acquisition of these 5 mutations in the genome, the fitness has increased to 1.35. So, there is a 35% increase in the fitness of the organism after acquisition of these 5 mutations. Let us quickly, I want to give a sense of what is the nature of genes that are targeted in something like, in a process like this. So, we will not get too much into the details of what the mutations were doing, but we want to list the names of the genes in which these mutations occurred. And the order in which they occurred in the experiment is the following. The first mutation to have occurred was in a gene called RBS operon. That is a first mutation. The second mutation occurred in a gene called top A. The third one, this was the second mutation. The third mutation occurred in a gene called SPO-T. Fourth one happened in the promoter region of an operon which drives the expression of GLM-US, and the fifth one occurred in a gene called PYKF. So, these are the 5 regions of the genome that were targeted in this specific order as the strain acquired these 5 mutations and became 35% fitter. 4 of these mutations are in the coding region, which means that if I have a gene, let us say this is the region of the gene, this is the gene, it starts with ATG, ends with a stop codon which could be any one of the 3, but let us say is TAA. So, in 4 of these cases 1, 2, 3 and 5, the mutation happened somewhere in the middle of the gene where a change in nucleotide happened. That is happening in these 4 cases. In the 5th case, something else happened where I have this 2 genes GLMU, GLMS, and they are collectively written as GLMUS, and the mutation that happened in this case happened in the promoter region. This region, a mutation in this region can often have one of 2 effects, that it increases the rate at which transcription happens by facilitating transcription factor binding or facilitating binding of RNA polymerase. Conversely, it can also reduce the amount of transcription that is taking place for the genes that are underneath this particular promoter. So, this region of a gene is called the promoter region. So, these are the 5 mutations that happened, leading to change of fitness from 1 to 1.35, a 35% increase. What we should also note is that a 35% increase is facilitated by acquisition of 5 mutations, which means that roughly each beneficial mutation, each of these is a beneficial mutation, each beneficial mutation confers a roughly 7% advantage. So, this is also the first time we are seeing a typical benefit associated with a beneficial mutation. So far all we have been saying is that a beneficial mutation occurs and fitness increases, but without any experimental data as to in a real evolutionary experiment what is the quantum of benefit that a beneficial mutation confers, and this is the first time we are seeing that data, that 5 to 7% is a typical number associated with a strong beneficial mutation that happens. Okay. So, as we did in the previous study, we are going to look at adaptive paths which are accessible or not accessible. In this case also, the same idea flows, that this is number of mutations, this is the ancestor which carried 0 mutations or the ancestor that I started with. This is the evolved strain which carried 5 mutations, evolved, As I mentioned that this experiment is something that has been going on for more than 80,000 generations, and many more mutations have subsequently been acquired by these strains. This particular paper was published keeping in mind only the first 5 mutations that got selected for in these lines. And here is fitness, which we mentioned was used by quantified by measuring growth rate as a proxy. So, our ancestral strain conferred a fitness of 1, let us say this is 1, and the evolved strain conferred a fitness of 1.35. And now, as we saw in the last example, going from 0 mutations to 5 mutations, there are 120 paths, and let us call those mutations A, B, C, D, E. So, again for those of you who did not understand that, how do we get these 120 mutations? When I am moving from 0 mutant to 1 mutant, I can pick any one of these 5 mutations, and let us say I pick B. So, that is my first mutation. Now, I have to pick the second mutation. So, I have 5 choices to pick the first mutation, out of which I picked B. For the second mutation, I now have 4 choices available to me, and let us say I pick D for the second mutation. So, I had 4 choices. For the third mutation, I have 3 choices available to me, and let us say I pick A, that is 3, and then similarly for the fourth one I only have 2 options available to me. This is my fourth mutation, and the fifth one, whatever is left I have to pick that. So, that is the only one. In all there are 120 paths. And again, if a path is monotonically increasing, 2 mutations, 3 mutations, 4 and 5, this type of a path is a monotonically increasing function, then this is called permissible in a Darwinian sense, permissible path. On the other hand, I could also have paths which look like this. In this case, after acquisition of the second mutation, the third mutation when it happened was deleterious because it led to decrease in fitness. In this case, this is a path which has a local peak, and hence it is non-permissible in a Darwinian sense. To understand a little bit more about why these local peaks arise, let us imagine that the order of mutations in the green path was A, B, C, D and E, which means this genotype was A, this genotype was AB, this genotype was ABC, this one is ABCD, and finally we get to the last genotype which is carrying all 5 mutations, it becomes ABCDE. On the other hand, in another path, in the blue path where the order of mutations has been changed, let us imagine that the order in which these mutations were acquired in the blue path is as follows. Let us say the first mutation to have happened was C, next one was E, then B, then D, and the last one was A. Now, what you should realize here, so this genotype here is C, this is CE, this one is CEB, this is CEDB, and the last one is the same in every path because last one is the same genotype where all 5 mutations are occurring. What you should realize here is, let us focus on mutation B. Let us see what is happening to, let us see what happens when mutation B occurs on the green path. On the green path, the mutation B occurs on a genotype which is already carrying mutation A. So, mutation A is already present on the genotype when B happens, and on this genetic background the new mutation B takes place. On this background B takes place, and as you can see here, going from genotype only A to genotype AB, fitness is increasing from this level to this level. So, acquisition of mutation B on a background where mutation A was already present led to increase in fitness, and hence on the green path mutation B, small b, mutation B was a beneficial mutation. But that is not the case for the blue path. Let us take a look at mutation B on the blue path. Now, in this case, mutation B occurs here when C and E are the two mutations that are already present. So, in this case, let us say C is already there and E is already there, and in this context mutation B takes place on this genome. But CE genotype had a fitness which was given by this number, but on this background when B mutation occurs, fitness drops significantly to this level. As a result, when mutation B occurs in a background which already contains mutation CE, fitness decreases dramatically, and in that context this mutation is a deleterious mutation. Deleterious mutation. So, what you should note here is that the nature of effect of fitness of mutation B is context dependent, and that context is dictated by what was the precise genotype in which mutation B was occurring. If it happened in one, it was beneficial, but if it happened in the blue path, it was deleterious, the same mutation. And this is of course the definition of sign epistasis, that a mutation is beneficial in one context but becomes deleterious in another context. And it is this sign epistasis which leads to these local peaks. So that is the reason why we have these paths like these blue paths which have this local peak, and the population would get trapped here. Back to our study of Cooper and co-workers, of the 120 paths, we are interested in knowing that how many paths belong to this category, the green category, and how many paths belong to this category. So, as with the previous study of Hartle and co-workers, this study also quantifies all 120 paths and tells us how many belong to the green category and how many to blue. And what they record is that out of 120, 86 paths belong to the green category and 34 remaining paths belong to blue category, which means that in this landscape most of the paths are adaptive in nature, that means they are permissible in a Darwinian sense, whereas a much smaller number, roughly 30% of the paths are not accessible via Darwinian fitness. Compare this with the previous study where about more than 80% of paths were not accessible and less than 20% of paths were accessible. So, that number of accessibility has increased to over 70%, and that is a huge change that has happened and the discrepancy that we note between the two studies. So, let us try and summarize this discrepancy here, that we have looked at two studies, each of them had 5 mutations. So, this was the 0 mutation variant, this was the 5 mutation variant. In both these studies, the 0 mutation variant was low fitness, 5 mutation variant was high fitness. Fitness was quantified differently in the two studies. In the first one it was MIC, in the second one it was growth rate. And MIC was Hartle and co-workers, and growth rate was Cooper and co-workers. And we know that in these two there were two types of paths. One was strictly increasing paths such as this. This is a permissible path. Alternatively, we had qualitatively different type of path which had a local peak. This is a non-permissible. A non-permissible path. And the relative fractions in Hartle, this was greater than 80%, and in Cooper this was less than 30%. And by the same token, for the permissible paths in the Hartle study, this was less than 20%, and in the Cooper study this was greater than 70%. So, the question then arises that how do we generalize this process and how do we understand what has happened here? In one case, we observe numbers which are like this. They tell us that evolutionary processes, these evolutionary dynamics process in a way such that a huge majority of paths are non-permissible. But in the other case, we get an answer which is the exact opposite of what we saw, which says that in a huge majority of cases, paths are permissible in a Darwinian sense. So, how do we reconcile this? In some sense, it is very fortuitous comparison where both the studies are doing the exact same comparison of 5-point mutations, 120 paths, and everything is organism is the same, and so on and so forth, but you get these diametrically opposite results. So, the way we think about this issue now is the following, that one big difference in the two studies was that in the first one, which was by Hartle and group, what was studied was one gene which acquired 5 mutations. All of this happened in one gene. In the second study, Cooper and group, the mutations that happened were in 5 different parts of the genome and in 5 different genes. Five different genes. And since then this problem has been studied in different contexts this model system, this was done here in one gene, but we have also then studied other genes to study this particular phenomena of the Huttle group. And what we now know, that when it comes to studying epistasis within a gene, sign epistasis is rampant. There is a lot of sign epistasis, which means that when a gene is evolving, the genomic context in which a mutation happens becomes very important. The same mutation in one context could be beneficial and could be deleterious in another context. And the way we should understand is that this entire piece of DNA is going to get transcribed and translated. So, we will get the mRNA, then we will get the amino acid chain, and this amino acid chain will fold to give the protein. However, if a mutation happens, this mutation in a folded state will interact with neighboring amino acids, and it is these interactions which matter. This interaction might be a good interaction, but if another mutation changed the amino acid right here to red, then this green may not want to interact with the red amino acid, and in that case it becomes deleterious. So, these interactions between proteins, between amino acids in a protein, because all these mutations are happening in the same gene, the gene when transcribed and translated the resulting protein has to fold, and these amino acid residues have to interact with each other. That is not the case that is happening here, because each of these mutations is in 5 different genes, and these genes, at least in this context, are not coming together and working together as a protein complex. They are all doing their independent things, not directly associated with each other. So, sign epistasis is not is very prevalent when we are talking of epistasis within a gene, but not that prevalent when we are talking of epistasis between genes. Instead, what we note in the 5 gene study is that negative epistasis dominates when we are talking of between genes. Remember, negative epistasis means that this is the effect of this is the fitness of the background So, let us say there are two, this is one background and this is another background, this one carries this one mutation, this fitness is F1, this fitness is F2, and on both these backgrounds the same mutation happens. Let us say on both these backgrounds this particular mutation happens. Now, when this mutation happens, this is a beneficial mutation, and on the y axis we will say effect of mutation. The same mutation happening, negative epistasis takes that says that it will have a greater benefit in a background of lower fitness as compared to the benefit that it confers on the genome of higher background. So, even though the mutation happening is the exact same in both of them, but their fitnesses are different, and hence the fitness benefit that this same mutation confers on the two is going to be greater in the background with lower fitness as compared to the background with the higher fitness, and that is negative epistasis. And these issues of epistasis, these properties associated with negative epistasis are what dominate, what is the dominant feature of epistasis when it comes to between gene epistasis. So, through these two studies we want to understand, we saw that epistasis plays out differently within a gene and between genes. And in the next video, we will continue with the third example, which is a more recent example published last year.